therapeutic area, if you can just assume what all therapeutic area we may have? Any idea? Very right. So cardiovascular. Okay, so we have CVS, cardiovascular system. So under that, heart attack, heart failure, all those comes under this. So first you need to see drug development process starts with the identification of therapeutic area. Okay, first you have decided therapeutic area, then you will decide the disease. Okay, for example, malaria. There is one therapeutic area that we call as infectious diseases. Okay, this is infection. Infection, corona. Yes. Very good example. And malaria, all infections, which are spreading through, many infections you might have heard. So this is the therapeutic area. First we have decided. Then we will, like for example, malaria. This is an infection. Who's spreading? Parasites. So this we need to, for example, and do second thing, that is a different thing. What kind of drug we are going to develop? That is different part. First we need to identify. Okay, we have to draw some thought in our mind. Okay, this is a therapeutic area. Then this is a disease. For example, infection. Then we have, for example, herpes simplex virus. Some viruses, all viruses, and bacteria comes under the infectious diseases because those are infections spreaded by bacteria and viruses. So very general example, you take malaria. Okay, I want to develop a drug for infectious therapeutic area and malaria disease. Two things are decided. So second thing, under drug development, what will happen? Third thing is drug discovery process. First will come drug discovery process. So how we can, like for example, some of you want to be.
Sir, how we can recognize that this disease has happened to the person before recognizing the drug?
Yeah, good. So for example, we have the audience. Audience, what I am telling you, see what kind of population, subject population we are going to target. Okay, first you need to keep in mind that we are going to target this. For example, what was happened? Corona. Suddenly this problem has come up. This they put into therapeutic area infection. So now, what was our target? Target population was the people who are infected with coronavirus, COVID-19. Okay, virus. So two things are clear. So you will see that okay, drug has not come into picture till now. Okay, drug has not come into picture. When drug will come into picture? Because we are going to develop drug. But the first step which starts, that is the identification of therapeutic area, disease. Then we will start identifying the other things. Now what will come into picture? For example, after post graduation, many people from here going to abroad for research purpose. Not saying here also we have some universities, there we have the research in bioinformatics, we have research in some other courses, microbiology. Okay, so these things we have research. So here actually because you all are here to get a job. Okay, you are looking for you all are here to look for a job. Now this first job will start in clinical research, that the students who are going from here to do some PhD or some research, they are going to they are taking admissions in universities. What these Pfizer and one more thing, see, in drug development, who all are stakeholders? Stakeholders, if I say, who are the parties who are involving in developing a drug? So here two things I would like to tell you. Hospitals, pharma industries, which I call sponsor companies. Okay. And third thing is what? Third thing is CROs. Example I will give you: IQVIA, Paraxel, and Icon, PPD. All these are CROs. These are contract research organizations. And about sponsor companies, example, very well.
Sir, then Cadila, Ciplas, and Pharma, Petrolabs, all these are companies.
Yeah, these are companies. See, these are pharma companies. For example, you have taken Cipla company. So I will not go deep into the companies, what is their structure, what they are doing. I will cover you Cipla, but now I would like to tell you, see, Pfizer, have you heard? Amgen, you have heard? Novartis, all these are sponsor companies.
And what is the difference between this company and this company?
Right, let me tell you. Cipla companies, Ranbaxy, you have heard two companies. So what happens, see, when drug will be developed, so there is a patent of 5 years. For example, one sponsor company, the sponsor company starts investing from this first step. The first step is, but they start giving some funds to the college, universities. So the students will come there, they will get fellowship, okay, scholarship they will get. So they will do some research. What research they will do? They will do research on the, for example, I have taken example of COVID-19. What they will do, they will start see, they have to you microbiology or some biotechnologies here, they will start thinking. You have to first you need to understand the pathology, how this problem is occurring. For example, COVID infection, you have to identify the structure of COVID-19 virus. You know very well virus is the we cannot make any drug for virus. Why? What's the reason?
We can decrease the number of infections.
Right. The problem is that, see, if somebody is having viral fever, so why I have taken example of virus actually, but I should take example of some other thing, because virus is actually, for virus we make the vaccines. There is a difference between drugs and vaccines. Why I am telling? Because virus actually, for example, virus is changing its structure after every, it may say 1 minute, after 2 minutes, after 1 hour. The structure is being changed. The protein's design is in a way that it is changing constantly. If something is constant, then only you can study that particular parasite, because that will remain same. For example, me. See, if I will take X-rays and all MRIs, so my body structure will be same. It is not getting changed. But the virus problem is that virus structure is getting changed. It is not constant. Okay, there is no stability. So if you will remove the protein from virus, the protein you will see, we have to, at this first step, see, I am going you all to the very deep clinical research, problem. And the first dope start with the research analyst. What these research analysts, what they are doing? Yes,
So
please.
if the structure of the virus is changed, so the amount of vaccine given to the person will
Yes.
be different in a
Yeah.
different form of the drugs.
Very,
Suppose
very.
it is 5 ml given to some person, and if the structure is changed, so different to the other person. So the ml will decrease or it will increase.
See, vaccine is nothing. Now, vaccine that infection, the same thing that extract of that particular virus we are injecting in your body. Your body will also, be adaptable with that particular. See, vaccine is nothing but that is the parasite itself.
Yes.
We are allowing our body to make antibodies. That you please remember that this is a virus, and viral fever, some viral fever, what they happen if they are not severe.
No,
So,
sir, I am telling dosage of the vaccine
yes.
will be decreased by changing the viruses form.
No, that will remain same. See, the step, if I will take you, vaccine also comes under the entire drug development process. This also, we have to do the clinical trials, phase 1, phase 2, phase 3, and that also, we have to see, vaccine is also, let me tell you, we also do the trials on vaccine in animals, preclinical studies. Clinical studies are done. We are making a vaccine, we are not making any such structure that will change. That will remain same. Our vaccine will remain same. But when we will inject that particular, see, at one instant we have identified that we cannot see COVID-19 first virus. When it was, came into picture. But when it is changed, the second vaccine is not designed. Okay. Second vaccine is not designed. If you remember, see, we are still identifying the different strains of viruses.
Sir, now it is recently come cervical cancer vaccine.
Right.
So it is given to a group of the age of only, not beyond that. Why?
See,
It is given, I think so.
yes, you asking
25.
question of the clinical trial part. Your all questions, like they are related to clinical part, because this is related to design. For example, clinical trials are conducted in the controlled conditions. Controlled, what I am telling you, controlled. See, if I want to conduct a clinical trial on X drug, for example, there is one X drug, I am going to conduct a clinical trial. So my target population will be fixed. Okay, I am taking 18 to 60 years of like aged group, and they should have some, we have some inclusion criteria, exclusion criteria. As per, that is why I am telling you. See, slowly I am going to that point. What you are actually asking, you are asking more about the study design. Okay, I will cover your this question in my part will come, but I am at the first stage. Let me just take you the story of clinical research. Because if, for example, if you're sitting in an interview, the panelist will be having good sound knowledge of clinical trials. They know at each step because you are just graduated, you don't have any practical experience as of now. What they will ask you question? They may ask you question about the clinical trials, some basic things, how it starts, okay, how it will go. But your question is not good, and I will take your question when I will be explaining under clinical trial phases. Okay, so first I am at the stage of the research analyst. What they are doing, the students in the universities, what they are doing? They are doing research. What research they are doing? For example, if I am taking example of some disease, for some cancer you can take, okay, you can take colorectal cancer. Okay, so what they will be doing? They are studying. What they are studying? They are doing real world evidence clinical research. What it means? Real world evidence. No problem, I will take you that. So, for example, colorectal cancer, many sponsor companies in Europe and USA and all other countries, there might have some in China also. Many trials might have been already conducted. Okay, all they have already like studied many things already about colorectal cancer, and there might be chance that there is one particular active compound.
What is that cancer? Can you elaborate?
Yeah, colorectal cancer. This is our cancer, colorectal cancer. You can study rectal, rectal part. So that is related to that part. So you have to first identify. See, I'm just taking you. Don't get confused. First, you need to identify the therapeutic area. Okay. Second, you need to identify the disease. The third step is what? You now start collecting the information about that particular disease. Whether how many clinical trials have already been conducted. There are different companies. For example, there are some companies, if you know, some Pharma Healthcare is one company. What that company is doing? That company is covering the journals. And also one more thing you should remember, each sponsor company has their own register, online registry. Whatever trials they are doing, all those clinical trials they are also publishing on their clinical trial register. And why I'm telling you about this, because this is linked with our first step. And one more thing, after conducting a clinical trial, when you said na, like phase 1, phase 2, phase 3, the phase 3 trial is registration trial. Means after completing phase 3, we have to file the application for marketing a drug. Okay, phase 3 trial. After completing these phase 3 trials, we are submitting the application to the regulatory authorities. Okay, so regulatory authorities, now a question has come up. What is a regulatory authority? So in each country, if somebody is doing clinical trial on the population of that particular country, okay, and they have completed phase 3 trial, so they have to submit the entire data to that regulatory authority, and that regulatory authority will study that data, and then what they will do, they will decide whether we will give permission to market a drug or not. Okay, so this is the regulatory authority. So you should remember, during the entire life cycle of your journey in clinical research, you should remember USA has one regulatory authority which we call as Food and Drug, FDA, Food and Drug Administration. And European Union, 30 countries comes under European Union, in all European. Okay, so what is that? What is the regulatory authority of European? Yeah, different. Swedish also. So we have like Switzerland, they have one. But EMA is taking care of all these. EMA, European Medicine Agency. EMA is just taking care of all regulatory part. Okay. So what is? They also have their own register. Register, again I'm telling you, what is the register? Online digital diary where all clinical trials conducted in the European Union, in the like, and some example, why we submit it to the USA FDA. We want that in India some drug is, we say for example, our Bharat Biotech, they have designed this, one vaccine. So that vaccine, if you want to that we want that we should market that vaccine in the USA, in the United States also. But we also need to submit the application to the FDA. to take permission that we can market this particular vaccine in USA as well. Okay, so that I will not confuse you. Just first thing, again, just repeat. First thing to identify the therapeutic area. Disease is with us, colorectal cancer. Third step, where we are. Now I'm telling you from where we should start. It's not directly that colorectal cancer. We will take some tissue outside. We will just start, extracting proteins and start identifying. No, we will not do that. Because why we will not do that? Directly we will not
Sir,
start.
diseases are how we can identify the diseases from the test and all.
Yeah, see, for example, I'm telling you, colorectal cancer is there. Oncology is therapeutic area. Now, how, when we are thinking that we will start developing a drug, okay, for this particular disease, from where we will start? Anyhow, we have to take the sample tissue. What will you take in cancer patients? What we'll do? We take the tissue sample, we study it, we take like what is the causing agent, like which protein, what like this comes under bioinformatics also and biotechnology. All these comes into picture. They start microbiology also. They start behind which, yes please. So which protein is causing this to see what is in our body. Every disease is what in our body, what is involving in every disease or health problems? Proteins. Either it is DNA, RNA. See, DNA making RNA, RNA making proteins. That is a simple thing. And proteins, we have 20 amino acids. In DNA, we have 4 ATGC. You know very well. Okay. So all these combination are only making our DNA genetic part, and also they are transcribed and transcription. Finally, what it is making? Proteins. Protein is only involving in all the process, enzymes and all those things. So we just need to identify what is the causing agent, what causing this particular problem. That is, but before doing this step, so we directly cannot take tissue and we start identifying. For example, if you did the same, and many other companies previously have already worked on this. So is there any use to do that again and again? For example, they have taken tissue sample, they have identified. Blindly, if we will start doing that, so we will also doing the same, and that was finally failed. For example, one clinical trial already conducted that was failed after phase 1, 2, 3, 4 pre-clinical studies, clinical studies phase 1, 2, 3, 4, everything done, but that was not because permission was not granted by the regulatory authority. So it means what? It was failed. Okay, that particular clinical trial. So is there any use of doing the same? What we will do? First, we need to do the real world evidence. Real world evidence is what? First, we need to identify. For example, in India it is not happening, but in some good hospital it is happening. We have had your medical history, any medication if you are taking. So all this data they will take. Do you had any problem you past? So they will take information from you. They will write this electronic health records. You will take the course and you will be okay. So if there is any problem, like for example any adverse effect, you might have observed. So you will go to doctor that because of this medicine I am having some severe acidity problem. So you will write down that thing. So that drug is already, see, we are developing drug in 4, 5 or 10 years according to you, but it is not actually fixed. In 10-year trial will be conducted. That depends upon the type of study, a type of disease. Okay, COVID disease, in 1 year we completed the entire clinical trial. In 1 year, we were having only the 1 year time because COVID, that pandemic, what was happening.
In special cases, we can do like
yes, that is why. So it depends on the study.
It
It depends on what? Therapeutic area. It depends on disease. It depends on the design of the study. Okay, whether what we are doing. For example, studies are not fixed actually. Studies, it's not like every time we are creating new drug. It doesn't mean it's already one drug is in the market. We may checking that for other indication as well, and we may also checking that if we increase the dose, if we increase the frequency, so there are different design of the study. Yes.
Why the COVID vaccines are given after the interval of 3 times?
Yeah, because it depends on, see, I'm at the that research actually what you doing in the research laboratory, like we have the two I will take you the preclinical stage. That is my next step actually. So in preclinical, we study in the animals. And animals, what we study, like safety, toxicity, carcinogenicity, mutagenicity, okay, and efficacy also we check at some instant. For example, you're asking question actually in advance. The question I have remembered, so I will cover that part in this. But you please ask question because so that I may cover those in when I will be covering other part. I am at the first step. Drug development has 3 types. First step is drug discovery process. Second is preclinical stage. Third is clinical stage. You are looking for job in clinical research, so you are looking job in the third step of the drug development process. Okay, there are 2 other steps also, preclinical and drug discovery process. Now what I'm covering drug discovery process. Okay, and you ask the question why we make that 3 times or 4 times. Okay, so this is we are on the first step. I will cover it in the preclinical stage, and good question. So first step where we were, that we need to identify the record. What all information about that study. Now go to the, if there are 100 companies, they are handling their own registers, clinical trial register, and there are Japan has their own regulatory authority, India has its own. Okay, what is the India FDA regulatory authority? Central Drug Standard Control Organization. Okay. Yeah, Drug Controller General of India, DCGI. Okay. So see, every country is also maintaining their own register of clinical trials. Now it is difficult to go to 100, 200 to registries, check that how many trials conducted on this disease. Now you will say that, so some companies are like what they doing, they are collecting the data from all the journals, from all the registries, that we call as data mining. Okay, as you know that you are in the era of data science, data analytics. You might have heard that this is the time like data is becoming more jobs are in data field. Why we are saying that? Because of this. Because the first step in clinical research is also to handle the data. See, so many registries. So there are so many companies, those who like they are maintaining their own database. They are collecting clinical trial information from all the sources about that particular clinical trial. They are just maintaining in their own database, and they are mapping that. They are doing mapping of that. Mapping, what I am saying. See, if some information you are just collecting. So if for example there are 100, 2000, documents are there, you will say someone that you take this one. I have collected from 100 website, 100 registries. So you take this data. What that person will do of that data? Is there any use of that data? No use, because they cannot go to 2000. But that company which was collecting data from different registries, so they were doing mapping of that. As soon as new data was coming available, so they were doing mapping. Okay, at this moment this happened, and they have designed in a very beautiful way, so that it will take just hardly 1 hour to understand the entire story of one clinical trial or one drug's development. Okay, it will take hardly 1 hour for the sponsor company. For example, sponsor company will decide based upon that data. Okay, I want to, for example, if one sponsor company, how they decide that we will, your question, how they will decide that which clinical trial we should go for. Well, fever. Everybody knows paracetamol and all, so many medicines are there, they can easily handle. So why new company will try to do the same thing? They will try to novel drug, novel disease, the new disease. Okay, like for example, they will first see that, okay, for example, antibiotics. What is the problem with antibiotics nowadays? Bacteria, if you are taking regularly antibiotics, so bacteria become resistant. What will happen if, for example, if one person is taking regularly because of some problem, for example, diabetic patient, if they got some injuries, so they have to take regularly the antibiotics. What will happen if all the antibiotics? Any question online?
They will become drug resistant.
Yes. So drug resistant is very big challenge for us in future. So what companies are thinking? They are thinking that, okay, what is the scope? Because companies are there for profit. Okay, some foundation are also there. Some like for example, Clinical Research Foundation. For example, Bill Gates, Melinda and Bill Gates, foundation. We have one foundation. Some foundations, they are not working for profit. They are working for the welfare of the people. Okay, we want to do something. For example, Bill Gates, one foundation institute, and likewise we have many other institute. NIH, they are funding. In USA, we have National Institute of Health. So they are funding many clinical trials for the welfare of people. That okay, because who will think about that? But some companies, all pharma companies, they think about their profit. So what they will do, they will decide that therapeutic area, that disease where they have more scope. For example, if there is one particular disease, we don't have any therapy, any like a good therapeutic, like any good drug market. We don't have any good intervention. We call it intervention. We don't have any good intervention for that particular disease. So they will decide on that okay, we want to work for this, so that in future we may capture the market, because anybody suffering from that particular problem, they have to take that drug. Okay, that is the point of view of the sponsor companies. So research, I'm covering the research, the drug discovery process. So first they will go in all the data that we call as real world evidence. They will use the electronic health records. They will take use of journals. Okay, all that information. Some clinical trials are being conducted on that. Do you know that some clinical trials, if you will go in industry, so there are clinical trials where you no need to enroll patient. What we do in clinical trial, we enroll patients, not subjects. Phase 1, 30 people. Phase 2, more than 300 people, 1000 people in phase 3. We have to enroll patient, but there are some trials where we do not need to enroll any patient. Some trials are there, and
Phase
you said
zero,
and they also have phase 1, phase 2. Likewise, see, those are observational studies. Those are not interventional studies. Those are observational studies, because we are just doing the observation rather than we can case studies, case cohort studies. We can just go online in electronic records. We can use the data. Okay, is this good to do that? Yeah, we need to always set up a clinical
Sir, if
trial.
something happens in the trial, so who is responsible for all that? For the
That I will cover in clinical part. Yeah, I will cover everything. This
no,
also.
sir, this topic.
No, this is research part. We have not enrolled any patient as of now. What I am doing, we are just sitting in universities. We are doing research. We are research analyst, research scientist, research analyst. What we are doing, we are just checking. For example, we have taken the. For example, I am interested. See, based upon the interest, you will enroll in any program, PhD program. What is that PhD program? 5 years. What you will do? You will help some scientist in completing one goal. What is that goal? They already know that okay, I want to work on this particular area of interest. What is area of interest? Like for example, any disease. They're working. So they will be looking for one candidate who is interested doing research on that particular part. Because there are students, they have their own mindset that okay, I want to work on HIV, okay, disease. I want to work on this particular cancer disease. So there are different interest. Okay, I want to do research in this particular topic. And what sponsor companies are doing? See, sponsor companies just paying for the activities. You will see they are not involving in anything, but what they are doing, they are just managing the things. They are giving money to the professor in universities. They are hiring research scientists, research analysts. You have done masters, so you know basic how to do some microbiology, do some test here, something. So you under their guidance you will work. You will extract the protein. You will do the mapping of protein. What is mean mapping? Because 20 amino acid, I told you, all 20 amino acid making the 2 million protein. Everybody is different from each other because of protein structure. Everybody has their DNA, different set of ATGC or sequencing, which what we call it gene ATGC, gene sequencing, and proteome. Okay, protein sequencing. So we are checking the pattern. So what we do first, we extract the proteins. We are doing with the help of bioinformatics tools. We are just taking the alphabetical that sequence, how it is aligned. What we are doing, we have now software available in the market for bioinformatics tools. So we are just taking that protein into in the computational those softwares. So how the structure will look like? That will look like the three-dimensional model. You said na, like how we will identify. Now the question comes up. For example, if colorectal cancer example I have taken, so now what we will do, that microbiologist, yes, some other student, chemistry, industrial chemistry, something, they will go in different labs. The lab part that we call as in vitro. That is called in vitro. In vivo means inside the cells. In vitro means inside the test tubes. So research scientist, research analyst, what they will be doing there? They will be identifying like proteins, taking extracts, and they are taking gene sequencing. They will see that how it is different from the healthy people. Okay, they will compare it with the healthy people gene sequencing and how it is different. Whether it is muted, yeah, some mutation has happened, yeah, something. What is causing cancer? We say that UV rays. Sometimes we say some chemicals. Sometimes we eat something toxin. All these causing agents. So they are identifying this is the normal, normal healthy people, that protein, and this is the like infected protein. Yeah, but we compare
So
from
protein will be accepted from the healthy person also.
yes. See, they have data, na? Like for example, we are doing clinical trials, phase 1, phase 2, phase 3, 4.
So sir, every time it might be not same, no?
No, what I am telling you, see, what we do, for example, if we are doing phase 1 clinical trial. In phase 1, usually we enroll the healthy volunteers. So in that, there is one document is provided to them. We call it informed consent form. What it means? That we are telling them that after this clinical study, after you will take medicine, we will take your some tissue culture. Use some tissue, that culture we will take from you. We will preserve it for the future clinical trial. And that is written in the informed consent form. A person sign that okay, I am ready that you preserve it for 3-4 years, whatever, because they have plan in future that we want to research it, that if healthy volunteers, so they want to there are so many things actually, and involved with this, but at this level I am just taking you to the entire story of that how it is starting, because you are here, you will go for interview. So if all these things will be clear to you, you will be knowing that okay, we are applying for clinical research. Clinical research is not just one part, it is the drug development process. Drug development is what? Drug discovery, preclinical and clinical. They may ask questions to you preclinical also. It doesn't mean that they will ask you clinical research related question, because drug development involves 3 steps. The first step I have covered you, that what is the steps in that identification of therapeutic area, then disease, and then third thing is we are working on the identification of target. Target identification is the third step. You have already, but before that, make sure the third step is to go back and find out the information with the help of those companies. Sponsor company will contact some other companies who are maintaining the clinical trial data about one indication. For example, malaria. So far, how many clinical trials conducted? 1,000. So all 1,000 clinical trial information you can get from one company. Okay, this company is providing us everything, and they have designed it very beautiful manner. We can understand the data, and that is data science. How they have, like data scientists, what they are doing? They have huge amount of data, and they are mapping it in a very beautiful way with the help of some graphs, charts, tables. Okay, so with the help of that, they are just designing that. And that data, what they are doing with that data, the sponsor companies in this first step of drug development process, they are taking benefit. They are paying a good amount of money to those companies, because that is a requirement, and they don't have time to go back in the research publish journals to find out that information. So they just pay something to that, and they get information. They hand over it to the scientist. They search it for that how many, like for example, if one person, there are 4 ways to go to Bangalore from one location. From Hyderabad, there are 5 things to go. Everybody has already gone there. So we need to identify the new way to go to Bangalore. So they will work on that new one, not on the 5 already, like explored by some other people. If something is already explored, there will not be any resources available there. Okay, just imagine if some area is there, if some under the sea, under the over the in space also, if somebody is going to moon, there. If one person has already explored, so there is no use of going there. We can just ask that person that who has already visited there, so they can provide us data. So that is why the data so far, whatever work has been done in this particular area, so we will take that information from the company who has already, like those who have already worked, and with the help of this other pharma company, Informa Healthcare. So this is their database design, they are collecting information. So this is the first step. You have identified two kind of jobs now: research scientist, research analyst. The good job is that after doing 5 years, if successfully you have identified something new, you have handed over it to the company. So your professor, under which guide, that guide, that person will create one thesis. You will create one thesis. That thesis will be published, and that is the one task. You will be out of that picture. Either you may again get some job. Now you are doing PhD, but now you can get some other job as a guide in the same project, because you have in the same, for example, in that particular therapeutic area, you have worked for 5 years. Sometimes it takes 2 years to just extract the protein from the sample. Sometimes it takes 2 years time period to extract the protein from the sample, because it is very delicate, it may break. But in the desired format, if you want, so you need to repeat it, I don't know how many times. The sample is very less, you cannot take again and again tissue sample from the any person. So that is the first job I told you, because all these things, in my purpose and our purpose here, what is the purpose? To make you ready for the opportunities in this field. So that is why I will cover all the opportunities. Then we will zoom all these areas in upcoming sessions. What we will do, we will zoom, all these areas. We'll give you option that this is one area. For example, regulatory affairs, for example, CDM, for example, PV. So I will cover all these things in detail in coming sessions. And now my purpose in the first class is to understand what all roles we may have. There's many roles actually involved. I will cover all those roles. And but I think what's the time of class? I think 1 hour class. No? So okay. So let's cover it. At least first part I will cover today. Research. This first part, drug discovery process. Tomorrow I will take you to the preclinical and clinical process. Okay. The first where we were. If anybody would like to reiterate what we have done so far, how many steps we have covered today?
Identification of the
Disease.
then drug
Therapeutic area first.
therapeutic area,
Disease.
then drug discovery.
Third is to go into the literature and do the real world evidence based observations and to take the data. And then fourth step is what? To start work on the new targets. And now target will be so many, because I told you so many proteins are 2 million proteins are in our body. Now if for one particular disease there might be many target. So what we will do, we will extract the protein, that is the in vitro. In test tube we will extract the protein. We will take that sequencing of that into the computer, and in computer we will do the modeling, that we call as drug designing modeling. So how drug is designed? So drug is designing is the first step, and bioinformatician will come into picture. They will take sequencing of proteins, and they will make a very good nice model. And now they will see the target. That what the target if they have identified. For example, there are 4 types of receptors. Receptors are there. Like for example, these are pockets. Pocket, like there will be some pockets kind of in receptor proteins. There will be pockets kind of spaces will be there. Now what is we have identified, okay, so we will focus that, okay, this particular protein, if we will inhibit the action of this protein, this may be beneficial to to stop the growth of these cancer cells. Okay, that is the target identification. But in this pocket, now the target is what? So here you can write down definition of target. So target is what? Natural.
The target of this target.
Yeah, this protein which we have extracted, the pocket we have identified with the help tools. And target will be this protein only.
So heading will be.
Yeah, this is the target identification. What is that mean? What is this target? So target is natural existing molecular structure. natural existing molecular structure.
Existing.
Yeah, existing molecular structure. Existing naturally, which is existing naturally. So naturally existing molecular structure that involves in the pathology of interest. Pathology of the interest for which the drug is in development. For which the drug is in development.
After involves
Yeah, so I'm repeating again. So natural existing molecular structure that involves in the pathology of interest. Okay, pathology of interest for which the drug is in development. You missed something? Where you are? Okay, so you have, this is what? This is target. Target, you from this definition you can understand target is just a natural, because cells make tissue. Okay, the molecular structure. This is making a molecular structure. So target is, we have identified now target, and definition you can say natural existing molecular structure that involves in the pathology of interest. It should be clear that this particular protein is involving in the pathology of interest. Means this particular protein is causing the disease. If you will hit some other protein which is not causing the problem, that doesn't mean you will hit, cannot create anything for that. So you first need to identification of the target. That this target, if you will, we will kill this particular target. Thus we will stop the action of this protein. We will inhibit the action of this protein. Then we may stop the disease. Okay. Now next step is to identify the chemical compound. Okay, the chemical drug, the target, candidate, that new chemical, what we call new chemical entity. NCE, new chemical entity, we call it. Here we call it against target. This is the
lead compound.
potential candidate, chemical compound. So chemical compound is the main bit, because every drug is what? What is every drug? It is a chemical compound. It is a structure of, if you see the design structure, so it is a combination of chemical compound.
Sir, what is the heading? To identify.
This is to chemical compound identification. So you can the new chemical entity, you can just say identification of drug. You anything you can write down. No? So this is. So what is a drug? So this we will say chemical. This is chemical compound that has pharmacological. You can write down what it means. Chemical compound that has pharmacological and slash you can say biological activities. Which is likely to be therapeutically useful. Which is likely to be therapeutically useful. Okay, so I have given you two definitions. One is the definition of target identification. So you know now that what is the target. Now you know that against target, what is the chemical compound? What is the candidate? Our active compound, active ingredient, which we call as drug. Because we are doing drug development. Under drug development, the first step which we are covering, that is drug discovery. Okay, how many steps we have under drug development? 3 steps. The drug discovery, preclinical and clinical. Okay. So now you have covered today, and how many jobs you have identified in this? Because for us, research analyst, sometimes we call it research analyst also, research scientist. Okay, and what is the scope of this? If you are research scientist for 5 years, you will get one thesis published, and you will be in guide professor in the universities in future. You will then, under you, there will be many research scientists working under you. So this will be the scope. Okay, I will cover all this perspective also, because just to give theoretical knowledge it doesn't work. So you have to decide after this course that where you want to go and what is the interest. Okay, so you have good scope. We have scholarship available.
Sir, sorry to just.
No issue. You please.
Sir, what is the main interesting in all that? We have the 6 domains.
Yeah, that question is also very like early you're asking. We have 6 months also, so we have very ample amount of time to discuss about all these things. But I think in one day only if we
No,
cover
sir, from your point of view, I'm asking.
that question, I can't give answer because everybody has their own interest. Okay, personally, if you ask, then I can give you that. Okay, from this way, if you will go, so you will get finally this opportunity. And see, in all the areas, every area is good, but every area has their own pros and cons. As usually, everything has pros and cons. So pros cons, what I'm telling, in some you may get good opportunity very soon. In some you may get feel like after 10 years, okay, it is a totally headache. I want to quit this job. This is not good after 10 years. So that depends. Like I will cover all of these things, okay, practically. And so today, first you just remember this is the first class. Don't be in hurry. But if somebody has interview, so that is different thing. I may guide, okay, like, and also like the course is 3 months or 2 months. But if you are applying somewhere and you get a opportunity, you can contact me. I can guide you about next steps also. But otherwise, in one day we can't cover everything, and we have so many things to cover. And I will cover, and I will make you people like feel like okay, you have good experience also. It will be like practical exposure. I will try to make you people as as practical like also you will go through that. So that is why in very detail the first thing I covered. Now you please ask me question, online people, if you have any questions about the first step, drug discovery process. I think at this level this much should be enough. Otherwise we have 100 pages to 2000 pages books also available, but that doesn't mean for you people. Okay, that is like if you will start doing some research. Because one more thing, if you are opting for this research scientist, so you cannot even so many therapeutic areas, and you cannot remember how many diseases we have.